Hi students, we are talking about body fluids and circulation. Today's topic is coagulation of blood. When a blood vessel is injured, it activates a complex hemostatic mechanisms which involves some events. This results in stoppage of blood. So this is called hemostasis. To prevent further loss of blood, according to the magnitude of injury, so this entire clotting process is completed within 3 to 5 minutes. So this process or these hemostatic mechanisms involve some different events, some different mechanisms, like we just classify them into 3 different mechanisms. So let us study in detail all this. Let's start. So first, the first process is vasoconstriction. This is the first mechanism, vasoconstriction. So let us study what exactly vasoconstriction. Fine, this is the blood vessel. When a blood vessel is ruptured, so this is a ruptured part of the blood vessel, and in the wall of the blood vessel there are smooth muscles. So imagine all these are smooth muscles, all these smooth muscles, because there is a layer. Blood vessel has got 3 different layers. One of the layer called tunica media is with all these smooth muscles. Here are the smooth muscles. All the smooth muscles are present. So injury to the blood vessel is a stimulation for the smooth muscles to contract. So when the smooth muscles of the blood vessel contracts, so this will decrease, or this will decrease the diameter of the blood vessel. So as a result, blood vessel gets constricted. So this is what is vasoconstriction. Due to constriction of this blood vessel, the blood flow is minimized. Sometimes the constriction is so strong that even it stops the blood flow in that area. This is vasoconstriction, one of the mechanism for stopping the the blood loss. Second is called formation of platelet plug. So this is platelet plug formation. So how this platelet plug formation is stimulated? So let us see how exactly this platelet plug formation is stimulated. So fine, this is our normal blood vessel which is injured. This is an injured blood vessel. Fine. So once the blood vessel is injured, when a blood vessel is injured, so I said the blood vessel is with different layers: tunica externa, tunica media, and tunica interna. So there is one layer which is called as endothelium. This endothelium of this blood vessel is with squamous epithelial cells. The endothelium is made with. So whenever this endothelium of the blood vessel is damaged, it exposes collagen. This is collagen. Once the collagen gets exposed, all the platelets that are present in the blood will stick to the collagen, and immediately they secrete some chemical substances which makes all these platelets to stick together. They become sticky and stick together. So all these platelets stick together. All the platelets become sticky and they stick together. So this makes a platelet plug. And this platelet plug seal the wound. Right? So this is how the platelet plug formation occurs. Still, the process does not stop. And further, in case of injury, right? So there is like a complex mechanism of formation of web of fibrins. Formation of web of fibrins. This is one more interesting mechanism for clotting of the blood. Fine. So what happens in this formation of web of fibrins? When once the ruptured blood vessel, so imagine this is in this picture, this is showing a ruptured blood vessel. When once the blood vessel is ruptured, it results in releasing of thrombokinase. So remember, the point here is thrombokinase is a complex enzyme. So this formation of thrombokinase is the entire complex formation is under the control of several linked enzymes, enzymatic reactions. So these are activated by several blood clotting factors. So one factor activates the other and so on. So as a result, it's a chain. We call it as a cascade. So formation of thrombokinase is a cascade of several linked enzymatic reactions. So finally, the thrombokinase is released. Now, when once this thrombokinase is released, there is a plasma protein which is called prothrombin. This prothrombin is produced from the liver, and this prothrombin is inactive. And this inactive prothrombin is converted into thrombin by thrombokinase in presence of calcium. Now, when once the thrombin gets activated, this active thrombin acts on one more plasma protein called fibrinogen, which is also inactive and is a soluble form. This soluble fibrinogen is converted into insoluble and active fibrin threads. So just to make you people understand, look here. This is our normal, the blood vessel. Suppose if it gets ruptured in this area, so I said all these fibrins are activated. So all these fibrin fibers form a network, and I said this fibrin is active and insoluble form. It forms fibrin threads, and all these fibrin threads will bind and seal that particular injured part and forming a fibrin like a clot. This is what we call it as clot. Now when once the clot is formed, from the ends of the clot it starts contraction after a few minutes. This is called clot retraction. This is called clot retraction. So after clot retraction, a fluid starts coming out from the clotted part. This is called serum. And the serum is plasma without fibrinogen. Plasma without fibrinogen is the serum. It doesn't clot. So this is what he called clot retraction. Now I said in the mechanism of the formation of thrombokinase, which involves different factors, they all act in a linked enzymatic reactions. So these all factors are 13 different factors. Like we have fibrinogen, prothrombin, thromboplastin, calcium, labile factor. Fifth factor is omitted. It has no significance in case of clotting of blood. Stable factor, antihemophilic factor, Christmas factor, Stewart factor, plasma thromboplastin antecedent, Hegman factor, and fibrin stabilizing factor. These all the 13 factors are involved in cascade. So you have to remember all this. At least first 4 factors you try to remember. Fine. Next, everybody might be knowing it. like vitamin K is required for clotting of the blood. Now you can ask me, what is the role of this vitamin K in the blood clotting? We never come across vitamin K here. So what exactly the role of vitamin K here? Listen, naturally, as long as the blood is present in the blood vessel, it is prevented from clotting because of heparin. Heparin acts as anticoagulant. Heparin is an anticoagulant. As long as blood is present in the blood vessel, it will never clot. Heparin acts as anticoagulant. Fine. Now, whenever the blood vessel rupture, as you know that, the process of clotting starts and blood clots. So what is the role of this vitamin K? Look here. Vitamin K is required for the formation of factor number 2, prothrombin, factor number 7, stable factor, and factor number 9 and 10. These are the factors for their formation. Vitamin K is required. So in absence of vitamin K, so there are no clotting factors, so it prevents the clotting of the blood, which is called hemorrhage, blood loss. So for that reason, it is commonly called as anti-hemorrhagic vitamin. Fine. Next, one more question whether you students will get every time. Like how blood is prevented from clotting in blood banks when we collect the blood. There we are adding oxalates of sodium and potassium. What is the role of this? This oxalates of sodium and potassium acts as calcium ion removers, because we have discussed that calcium plays a major role in blood clotting. So they act as calcium ion removers, so prevent clotting of the blood. And also in blood banks, blood is stored by adding EDTA, ethylene diamine tetraacetate. This also acts as calcium ion remover. So as a result, the process of clotting is prevented. Right? So if you understand the lecture, give a like, share and subscribe, and press the bell icon to get notifications whenever I post a new video. Good luck.